RNA detection. This webinar is part of the 2021 Microbiology Virtual Week, and I am Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Millipore Sigma. For more information about Millipore Sigma, go to www.sigmaaldrich.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, just click on that Ask a Question box located on the far left of your screen. As a reminder, this presentation is also educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right-hand corner of your presentation window and follow the process to obtain those credits. Without further ado, I now present today's speakers, Nicolas Caffarelli and Dr. Saito, both R&D scientists at Millipore Sigma. For a complete biography of our speakers, please visit the Biography tab at the top of your screen. Nicolas and Khan, welcome to both of you. You may now begin your presentation.
Hello everyone, and welcome to today's LabTools webinar. I'm Nathan Nee, Associate Science Editor for The Scientist, and I will be moderating our discussion. Today, our speakers Nicolas Caffarelli and Saito will discuss new RNA extraction and purification strategies and how they enable reliable and sensitive viral detection. We'd like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and our speakers will address these during the Q&A session following the presentation. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. We will try to address as many of these as we can during our Q&A session. This webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsor. Millipore Sigma is the U.S. life science business of Merck KGaA, Darmstadt, Germany. Our purpose is to solve the toughest problems in life science by collaborating with the global scientific community. And through that, we aim to accelerate access to better health for people everywhere. We provide scientists and engineers with best-in-class lab materials, technologies, and services. We have a broad portfolio of 300,000 products, an expanded global footprint, and are dedicated to making research and biotech production simpler, faster, and safer. Our sponsor has provided us with some helpful resources related to today's webinar, and we have posted these in our handout section located on the right side of your screen. You can access and download these documents at any time during the webinar. With that, let me introduce our speakers. Nicolas Caffarelli has led the development and commercialization of various novel molecular biology and protein mass spectrometry products within R&D at Millipore Sigma. He has previously worked at the American Red Cross testing human blood and plasma donations, and as an adjunct biology professor at St. Louis Community College. He holds a bachelor's degree from the University of Texas in Austin and a master's degree from Washington University in St. Louis, where he researched cell biology and virology. Saito develops new research tools for epigenomics and functional genomics research as an R&D scientist at Millipore Sigma. He earned a PhD in molecular biology from Tokyo Medical and Dental University and conducted postdoctoral research in Pierre Kinet's laboratory at Harvard Medical School and Tomas Mustelin's laboratory at Burnham Institute for Medical Research. Saito has researched protein and lipid kinases, phosphatases, and calcium signaling in immune cells. He has recently expanded his focus to gene regulation and epigenomics. Let's just make sure everything is set up properly All right, the floor is yours.
All right, so thank you all for joining the call. My name is Nicolas Caffarelli. I'm a R&D Scientist at Millipore Sigma, and I'll be giving the first part of this presentation. Before we get started, since there may be folks listening in from different parts of the world today, I think it's important that we clarify that within the United States and Canada, we are known as Millipore Sigma. However, everywhere else in the rest of the world, we are Merck KGaA. Okay, so for my part of the talk, I'm going to begin by giving an introduction on COVID-19, which I'm sure we're all very familiar with, followed by a little bit on SARS-CoV-2 biology. This is the virus that causes COVID-19, and then a quick primer on the qRT-PCR technique. Afterwards, I'll jump into GenElute, which is a kit that we offer for column-based viral RNA extraction, and some data that we've gotten with that kit, looking at SARS-CoV-2. This will be followed by an overview of a new product, Viral RNA Extraction Buffer, which allows for quick, convenient lysis of virus such that it is qRT-PCR ready, and then close out with a summary of findings. So, jumping right in. Okay, so as we all know, COVID-19 has been around for a little over a year and a half now, and really represents a serious global crisis. As of this month, there have been over 170 million confirmed cases and more than 3.7 million deaths reported. However, fortunately, vaccines are becoming more accessible globally, and it is estimated that over 2 billion doses have been administered thus far. But nonetheless, as you can see in the figure on the right, SARS-CoV-2 variants with varying degrees of concern have been emerging globally, with some of these having been shown to have characteristics such as higher transmissibility, more effective immune evasion, and or more disease severity. I should point out, as the authors note, that Europe and the UK are overrepresented in this map, not necessarily because the variant originated there, but because a lot of sequencing is done there, and so this is often where new variants are first identified. Another significant cause for concern, as shown in the model on the bottom left of this slide, is that current vaccines may be somewhat less effective against emerging variants as viral surface proteins mutate away from neutralizing antibodies. So unless we're able to completely eradicate this virus, we All right, so thank you all for joining the call. My name is Nicolas Caffarelli. I'm a R&D Scientist at Millipore Sigma, and the whole particle then becomes reassembled at the ER-Golgi intermediate complex. The result being in particles that are trafficked to the cell surface and exocytosed. And so the result of this is that you have a viral particle that is fairly dense in membrane and surface proteins, lipid bilayer, which is host cell derived, and inside is the genomic RNA, which is tightly bound by this nucleocapsid protein. The genome itself is fairly well characterized. You have these protease-derived proteins that are believed to have a number of functions, largely involved in viral particle assembly. The polymerase and helicase, which help with RNA replication, and then these viral proteins, the spike, envelope, membrane, and nucleocapsid, and all of these right here are going to have abundant mRNA within the host cell, so they serve as sort of attractive targets for mRNA detection. They're represented both as their own individual mRNAs, but they will also be on strands of the entire genome. Okay, now I want to change gears a bit and just touch on one-step qRT-PCR, which is the technique that we use for detecting viral RNA. I think Con will go into a little bit more detail here, but I just wanted to give a quick overview. And so the way this works is that for a one-step QRT-PCR reaction, you put everything into one tube. So you'll have your template and specific primers along with both transcriptase and polymerase, and anything else you need for a reaction such as DNTPs, buffer, magnesium chloride. And then both the reverse transcription and the PCR are all built into your thermal cycler program. So you start with your reverse transcription. It's usually around 44 degrees where the enzyme is happiest for about 15 or 30 minutes. This is perhaps followed by hot start activation, and then you have your actual qPCR in which you cycle through and measure fluorescence at the end of each cycle. And so the result is that you'll get data sort of like what you see here on the right, where you have an amplification curve that is exponential as you have doubling of DNA in your PCR reaction, which then eventually plateaus off as you exhaust your reaction reagents. You can then set a threshold, a baseline threshold, that you want to put right around when, again, the reaction starts to become exponential. And where the amplification crosses this threshold, we designate that as the CT value. And so the CT value is more or less a quantitative indicator of how much template you had to begin with or how sensitive your reaction is. So if you have a low CT value, and this is all shifted to the left, that means your amplification started much earlier, so you had a lot more template, a lot more RNA or DNA in there to begin with. If your CT value is high, and this whole thing is shifted to the right, then that means that you have significantly less template in there. If you don't have any amplification at all, that would be considered a negative reaction, so you don't have any template detected. And so in this talk, I'll be showing QRT-PCR data of SARS-CoV-2 RNA. So we target the N gene using these two CDC-validated primer probe sets, and I use our one-step RT-qPCR kit from the Sigma Aldrich catalog. So this is product number QR200, and this comes with everything you need. It has the reverse transcriptase, magnesium chloride if you want to supplement in, which I do, and PCR ready mix that contains TAC, buffer, and DNTPs, so that all you have to do is add your primers and probe. QR200 also comes with PCR buffer as a diluent and reference dye if you need either of those, but I don't use them in these particular experiments. And so here on the right, you see an amplification curve of a dilution series of synthetic SARS-CoV-2 RNA. And this is specifically targeting the N2 gene. And so what you see is that I can detect variably down to 10 copies, consistently down to 100 copies in my hands. But as you'll recall, viral mRNA is not necessarily accessible for qPCR. Rather, it is encased in this particle where you have your lipid bilayer and your spike and other membrane proteins. So in order to do qPCR, you need to break open the viral particle. And the way this is most commonly done is using a column-based method where you have, for example, a first lysis that then involves binding to the column, washing out any leftover lysis solution, such that you can then elute off clean, purified RNA. We recommend adding carrier RNA at the beginning, as that can help with stability and yield, especially if you have small titers to begin with. And this process was adapted to our already existing RNA Miniprep kit by scientists in our organization, Kelly Wolf. And so you can find the protocol for this online, and I believe it's also included as one of the accessory materials for this talk. So when developing this protocol, Kelly used largely armored RNA, but I wanted to try it out in my own hands And so the experiments that you'll be seeing here is with cross-link inactivated SARS-CoV-2, which we get from Zeptometrics. And so the first thing I wanted to see is if this process is compatible in some different backgrounds. So I'm looking here at saline, particularly PBS, and then viral transport medium, which is sort of the most common medium for nasopharyngeal swabs when folks are doing virus testing. So I spiked these cross-link inactivated particles into either of these mediums, purified with our GenElute Miniprep kit so that we get purified RNA out of this, and then I put it onto our one-step RT-PCR kit. This is QR200. And so we see good amplification at two different concentrations, either 100,000 particles per mil or 2,000 particles per mil. As you would expect, you get higher CT value when you're putting less particles on there. And I did this with both N1 and N2 primers, and N2 is maybe a little bit more sensitive, but overall they both work well, and they work in both PBS and in viral transport media. And so we see that our GenElute kits work well for prepping viral mRNA. I wanted to dive a little bit deeper into this and see really how sensitive this was, how low could we go. And so I did two subsequent experiments diluting down virus based on the amount reported by Zeptometrics, these cross-link inactivated particles. And so I go down to about 125 or 250 virus per mil. Since I'm only loading 120 microliters of sample, that equates to 30 or 15 viral particles being loaded actually onto the column. And so what I see between these two experiments is that with N2, I'm able to get down to 250 virus per mil, or 30 virus per load. N1, quite not as sensitive. I get down to 60 virus particles being detected. But still, this is quite sensitive. And so you see here that this is fairly linear. And really, if we wanted to increase sensitivity, one thing that you could do is increase the sample load, assuming that you have a good amount of sample, because you are essentially concentrating on the column. So if, let's say, you do a 300 microliter sample load, 50 microliter elution, then you would get six-fold concentration, and that would potentially be quite sensitive. But for our purposes, we demonstrated that we could get pretty low, so we were happy with this data. But we do realize that column-based approaches are not without their drawbacks. So even though it's generally easy and straightforward, it does require centrifugation or manifold, vacuum manifold, so it can be a little bit time-consuming. And more importantly, these kits can often suffer from supply chain shortages. And we saw that this was really an issue sort of as the pandemic got underway, and it was a serious bottleneck for a lot of virus detection. And so we endeavored to come up with an alternative. And so earlier this year, we launched this product, which was developed in the fall of 2020, which is our Viral RNA Extraction Buffer. And so the way this works is it's a 4x solution. So you take, say, for example, 6 microliters of your sample, and you can add 2 microliters of viral RNA extraction buffer, and all it is a 5-minute room temperature incubation in which you lyse open viral particles such that they are directly ready for qRT-PCR. So this can actually even be performed in your qPCR strip or plate ready to go on the machine. One thing we did find is that if you're using saliva, you get best results centrifuging, and then taking the supernatant so that you don't have sort of debris and particles getting in the way of your reaction. And so this product has worked well. We find that, again, all you need is a 5-minute incubation. It's a crude prep, but there's no cleanup required. And we have some data demonstrating that it's compatible both with qRT-PCR and LAMP, which we'll show, and that also it's compatible with a number of different backgrounds, so things like saline, viral transport media, and saliva. So in developing viral RNA extraction buffer, we wanted to make sure that it was compatible with a number of different backgrounds. So in order to demonstrate this, what we're showing here is I took cross-link inactivated SARS-CoV-2 particles, again from Zeptometrix, and spiked them into either saline, saliva, or one of these viral transport mediums, lysed with viral RNA extraction buffer, product number VRE100. This was again a 5-minute room temperature incubation, such that the result is a crude RNA prep. So we're not purifying anything out, but the RNA is now accessible by the qRT-PCR reaction components. And so we put this again with QR0200, our one-step qRT-PCR kit. So if you look at our data in saline, again we did a dilution series of particles per mil, and we see, as you would expect, that as you have less virus, your CT value increases, but we are able to get down to 1,500 particles per mil. Saliva, not quite as sensitive, so saliva is kind of tricky to work with. It's highly variable between different people. Sometimes you'll have something that's really easy and kind of clear. Other times you might find saliva from someone that's viscous, full of debris. So we wanted to test it across different individuals. So here in this figure we show saliva from 4 individuals, all healthy. And so here we see that it's not quite as sensitive as saline. So we're detecting down to 50,000 viral particles per mil. However, this is relative to the viral load that you might see in a patient still pretty low. And we see that actually for 3 out of 4 people, we probably could have gone considerably further, but one of these had particularly difficult saliva, and so we didn't detect at 2.5 times 10 to the 4th, or 25,000. So this is sort of where we say our sensitivity goes down to. Similarly, we did test it in a number of other mediums. So again, viral transport media, other commonly used storage mediums for nasopharyngeal swab, such as eels or AMIs. And here you see that it works in all of these. We're looking at 1,000 viral particles per mil, 10,000 viral particles per mil. And if you don't treat it, you don't get any signal in these backgrounds. But if you do treat with viral RNA extraction buffer, then it appears. So this demonstrates that in order to detect at least this amount in these mediums, you do require at least some kind of lysis. So this was all very encouraging. We're very happy to see that our viral RNA extraction buffer seems to work well across different mediums. And we were curious sort of how it compared to similar approaches that, like viral RNA extraction buffer, don't rely on column-based purification. And so the closest thing that's out there that's sort of most commonly used from the literature is this Proteinase K with heating method. So in this experiment, I replicated sort of one of these commonly used protocols where I add Pro-K, vortex for a minute, heat, and then centrifuge. And then to the same samples, I treated them with viral RNA extraction buffer. So this is 1 times 10 to the 5 cross-link inactivated particles, SARS-CoV-2 particles. And again, this is in saliva from 4 different healthy individuals. And so what we see here is, on the right, a representative amplification, is that we get amplification much earlier, and so we have much stronger signal in our hands using viral RNA extraction buffer. The Pro-K plus heat method worked, but we didn't really get CT value up until the high 30s or low 40s. And then if you use neither method, you don't get any amplification. So this data demonstrates, again, that in our hands, viral RNA extraction buffer does give lower CT values, so it's a sort of more robust technique, and that also it's a little bit more uniform in the output. So you can see here that the data points are clustered a little bit closer together. So we believe that this is a little bit more reliable than using Pro-K with heat for virus detection by qPCR. So the last piece of data that I'll show today is the only one that doesn't use qRT-PCR as a readout. This is work that was done by Tony Anima, an R&D scientist, who set out to demonstrate that viral RNA extraction buffer is compatible with LAMP, or loop-mediated isothermal amplification. And so this is a pretty neat technique. It uses a strand and complementary strand that form these sort of dumbbell shapes, which can then amplify off of each other. And what's neat about this technique is that it's isothermal. So you don't actually have to do thermal cycling. You just maintain an elevated temperature where the LAMP enzymes are happiest. So I believe this was done at 65 degrees. And you can then get your readout. So for this experiment, Tony used the same cross-link inactivated particles that I had been using. And he lysed these with viral RNA extraction buffer, VRE100, so that he has crude RNA. But instead of going on to qPCR, he went onto a LAMP assay kit, a colorimetric LAMP assay kit that's commercially available from NEB. And so what you see from Tony's data is that as a control, he just looked at first water and synthetic viral RNA. So this is already exposed RNA, not particles. And you can hopefully see here that negative result has this sort of purple color. Positive result, so if RNA is detected, is this yellow color. And these are both done in the absence of viral RNA extraction buffer. So the next control that you see here to the right is using the same conditions, but in the presence of viral RNA extraction buffer. So again, synthetic viral RNA is detected by this colorimetric LAMP assay. It gives this yellow readout. If there's no template, then you have this negative readout. So this kind of pink or purple colorimetric result. So this tells us that viral RNA extraction buffer doesn't interfere with the LAMP assay itself. And then finally, within sort of the experimental group, if Tony runs viral particles in saliva through the LAMP assay, but without viral RNA extraction buffer, a lot of the RNA is inaccessible, and so you get a negative result even though there are viral particles in here. However, if you lyse first with viral RNA extraction buffer, then you do get your positive result. So this indicated to us that viral RNA extraction buffer is compatible with LAMP and can be paired with LAMP, and specifically can help you get results if the LAMP assay by itself is not enough. So a little bit of a sensitivity boost on this LAMP assay readout. Okay, so to summarize, today we looked at two different approaches for making viral RNA accessible for detection by qPCR or by LAMP. The first of these, a Miniprep using our GenElute mRNA Miniprep kit, we found worked well. Its advantages are that it's highly sensitive. The material can be concentrated, which would even allow for more sensitivity. And because you're getting a pure, clean prep, this is likely better for long-term storage, say in the freezer. And so with this approach, again, because it's very sensitive, we detect down to 500 virus per mil in viral transport medium. However, if you want something that maybe isn't quite as sensitive, but is quick, convenient, easy, amenable to throughput, we offer this new Viral RNA Extraction Buffer. So again, this is a 5-minute room temperature incubation, no column-based cleanup, and we tested it across various sample types. We want to show that it can be used broadly in different backgrounds. So detecting as low as 1,500 virus per mil in saline, or 50,000 in saliva, or 10,000 in transport mediums. So with that, I'll hand it off to Khan, and I'll take any questions at the very end. And I want to thank everyone for listening. Thank you.
Hello, I'm Saito, and I'm an R&D scientist at MilliporeSigma. And in the next section, I'll talk about the principles of one-step RT-qPCR assays, the differences between the kits that we supply, and the evaluation of the kits for SARS-CoV-2 virus detection. This figure shows the SARS-CoV-2 genome. SARS-CoV-2 is a positive strand RNA virus and is about 30,000 nucleotides in length. At the 5' end, there are two large ORFs, ORF1A and ORF1B, which cover over 2/3 of the genome and encodes non-structural proteins. The last third of the genome encodes 4 structural proteins, namely S, the spike surface glycoprotein, E, the membrane protein, M, the membrane glycoprotein, and the N, the nucleic capsid phosphoprotein. Because the genome is a single-strand RNA, mutations can be introduced during the replication process. In this figure, the lines represent the number of mutations found in 4,400 SARS-CoV-2 genomes. Within these mutations, the S protein mutations are especially concerning, since these mutant proteins can increase bindings to cell surface ACE2 receptors during host cell entry, which increases the transmissibility and severity of the disease. Variants that carry S protein mutations have been identified and classified as variants of concern. Here is a list of some of the notable variants that are classified variants of concern VOC or variants of interest VOI by the US CDC. B117 variant that was first detected in the UK is one of the most widely spread variants. This variant carries the E484K mutation, and its transmission is about 50% higher. Similar variants that increase transmissibility were identified, like the B1351 from South Africa, P1 from Brazil, B1429 from California in the US. And recently, the B1672 variant was detected in India. This variant carries L452R and E484Q mutations, and significant reduction in neutralization by antibodies generated during previous infection or vaccination were reported with this variant. Although this variant is currently categorized as VOI, it is already majorly concerning. Now let's go back to PCR detection of the SARS-CoV-2 virus. When the COVID-19 pandemic began, RT-PCR tests were the first to be developed, and they remain the primary tool used for diagnosis. The US and other countries developed assay targeted at the N protein gene. Because of the abundance of the N gene subgenomic messenger RNA, RT-PCR assays targeted the N gene could theoretically achieve enhanced diagnostic sensitivity. So previously developed MERS-CoV and SARS-CoV assays also targeted the N gene. This figure shows the sequence of the N gene. As you can see, several countries developed their assays targeting this gene. The US CDC developed N1, N2 assays at these locations. Here are sequences of the primers and Tacman probes for the N1 and N2 assay. Next, I will explain the principles of dual-labeled probe assay, or the Tacman probe assay. The Tacman probe consists of fluorophore covalently attached to the 5' end of the oligonucleotide probe and quencher at the 3' end. The quencher molecule quenches the fluorescence emitted by the fluorophore via FRET. In the PCR reaction, the primers and the probe anneal to their specific target sequences. The polymerase extends the primer and synthesizes the nascent strand. When the polymerase reaches at Tacman probe, its endogenous 5' nucleus activity cleaves the probe, separating the dye from the quencher, thus relieving the quenching effect and allowing fluorescence of the fluorophore. Now I'll explain the principles of the one-step RT-qPCR kits. In one-step RT-qPCR, reverse transcription or cDNA synthesis and qPCR are carried out in a single tube by employing sequence-specific primers for the amplification of the specific target. The kit performance are dependent on three components, namely reverse transcriptase, RNase inhibitor, hot start DNA polymerase. Reverse transcriptase is one of the most important components of the kit. Mononemurin leukemia virus reverse transcriptase, MMLVRT, is used in most one-step RT-qPCR kits. In the past few decades, the enzyme evolved through genetic modifications. The RNase -point mutation was the most common mutation, which increases reverse transcriptation activity and improves thermostability. In addition to the point mutation, multiple thermostable mutations have been discovered. Here is a list of one-step RT-qPCR kits we supply. The QSTART kit and ABScript kit employ MLVRT with both RNase -, mutation and thermostable mutations, and the RT temperatures are increased to 48 to 55 degrees Celsius. The next important component is the RNase inhibitor. RNases can be introduced from samples, primers, or probes. This figure shows one-step RT-qPCR assays with and without the RNase inhibitor. As you can see, there are 5 CT differences between the assays, meaning they differ by a factor of about 100. Finally, the efficiency of the hot start DNA polymerase is a critical factor. There are 2 different hot start DNA polymerases, namely antibody-mediated and chemically modified. In general, antibody-mediated DNA polymerases can activate in less than 2 minutes at 94 degrees Celsius, compared to chemically modified enzymes, which take 7 to 10 minutes at 95 degrees Celsius. All RT-qPCR kits we supply use antibody-mediated DNA polymerases. Here is a summary of the factors that affect the performance of one-step RT-qPCR. Genetic modifications of MMLB reverse transcriptase, RNase H plus or minus, thermostable mutations which affect the RT temperature, quality of MMLB reverse transcriptase, quality of RNase inhibitor, the efficiency of Taq DNA polymerase, and finally, the buffer compositions, including the magnesium chloride concentrations and the DNTP concentrations. When the COVID-19 pandemic began, we evaluated one-step RT-qPCR kits which we supply with the US CDC N1 and N2 assays, though those kits are research use only. Using in vitro synthesized SARS-CoV-2 genome RNA, we evaluated the one-step RT-qPCR kit using the following criteria. No abnormalities in the amplification curves. The R-square values and slopes from the standard curves are within range. No amplification from non-template control NTC samples, which is important to avoid false positive results. The CT number from a sample of 100 copies, smaller is better. The minimum copy numbers that kit amplify, lower is better. We evaluated these one-step RT-qPCR kits with N1 and N2 assays, and we confirmed that all amplification curves were normal and that the R-square values and slopes from the standard curves are within range, and there was no amplification from NTC samples with these kits. This table shows CT values from 100 copy samples and the lowest copy numbers amplified for both N1 and N2 assays. And from these results, all kits shows decent efficiency and sensitivity detecting SARS-CoV-2 RNA. Between the lowest copy detected is between single digit to highest number would be 100. Sigma-rich QR0200 quantitative RT-PCR ready mix kit shows the lowest CT number from 100 copy samples for both N1 and N2 assays. The newer Sigma-rich QStart kit shows overall best performance, like lower CT value and the lowest number of copy to be detected for both N1 and N2 assay. And Sigma-rich AbSwift2 kit follows. Finally, we tested compatibility of one-step RT-qPCR kits with VRE100 Viral RNA Extraction Buffer. The QR0200, QStart, and Kappa kit, as well as some Fisher's TaqPath kit, shows excellent compatibility with VRE100. To summarize, in response to the community's need for SARS-CoV-2 specific reagents, we have evaluated one-step RT-qPCR kits for SARS-CoV-2 target detection for research use only. All one-step RT-qPCR kits which we supply demonstrated excellent performance detecting the SARS-CoV-2 RNA. Our newest QStart one-step probe RT-qPCR ready mix kit, which utilizes the most innovative technologies, performs best overall, and the quantitative RT-PCR ready mix kit, QR0200 also performed very well in the evaluation. The QR0200 qPCR and Kappa kit, as well as some Fisher Tuckpass kit, exhibit perfect compatibility with VRE100 Viral RNA Extraction Buffer. Thank you for listening, and I will be happy to take questions after the acknowledgement. Thank you.
So,
thank you very much for those presentations. As a reminder to our audience, you may submit your questions to our speakers using the Q&A tab at any time. Our audience has already submitted several questions, so let's begin our Q&A session. And for the first question, I will address this to Nicolas first, and after Nicolas answers, if you have anything to add, please feel free to chime in. Our first question is, are the viral RNA extraction buffers or the qPCR kits, can they be used for diagnostic purposes?
Yeah, so Nicolas here. These are, as indicated, research use only grade products. So our organization, we don't develop diagnostics or IVD grade products. That being said, if a lab wanted to incorporate any of these products into a diagnostic workflow, it would be their responsibility to validate it within that workflow and to make sure that they are compliant with any applicable regulatory agencies. And we'd be happy to help as best we can towards that. But yes, ultimately, we develop them research grade. Any use in diagnostic beyond that would be the responsibility of the customer or of the testing lab to validate.
Thank you, Nicolas. Khan, do you have anything to add to that, or?
It's depending on the each country's regulations, so we cannot comment for this.
Our next question, and for this one I will start with you, Khan. What sort of sample contaminants could lead to amplification failure?
This can be various things. Can think some of the contaminated microbiomes, or if you contaminate with some of the blood content like hemoglobin, those things can inhibit. So from that point of view, the kit like column-based purification that Nick shows, GenElute, could be the best way to purify RNA for PCR sample.
Thank you, Khan. Nicolas, for our next question, we have several comments and several questions from our audience asking about some clarification for the experimental parameters. So I suppose we'll just get started with those. One question asks whether or not you tested the viral RNA extraction buffer using samples stored in inactivated transport media, ITM.
So the answer is no. There's a whole lot of different storage and transport mediums out there, so that's not one that we've gotten to yet. But it's a good question and a good suggestion. So I think it's something that we should look at, and hopefully we'll have some data for you in the near future on that. It's a good question.
Thank you very much. Next, for either Nicolas or Khan, were any of the experiments that you did where you presented the data in this presentation, were any of them done using pooled samples, and potentially pooled samples where individual samples were processed in different ways?
I didn't do anything like that. I think in theory it's something that you could try, but it's not something that we tested in particular, or that I tested in particular.
Yeah, it's possible to retest for some of the pooled sample and maybe run the NGS, not the PCR, but we didn't test.
All right, thank you. And the last question along this kind of line of questioning, were any of the samples obtained using nasopharyngeal swabs?
So the answer is no. Again, because we are doing sort of research use only products and we don't have a BSL setup or appropriate BSL setup at our facility to handle NP swabs, we tried to sort of, from a research standpoint, replicate that as best as we could using these cross-link inactivated particles and transport medium, as you might see in an NP swab. I will say that we are looking for clinical collaborators who are trying to see how this works in NP swabs. So if that's something that your lab is interested in and you're working with NP swabs and you'd like to try out VRE 100, please feel free to reach out and we'd be happy to send you a sample and work with you.
Thank you, Nicolas. So along that kind of line of thinking, do you anticipate any issues with samples that come from different sources or have different processing, or possibly more heterogeneous samples such as wastewater, for example?
Yeah, we haven't looked at wastewater. Certainly, I think the big takeaway here again is if you anticipate a sort of very messy mixture, a very complex sample, then for a very sensitive, reliable approach, we would recommend using a column-based method. If, however, you want a quick, convenient screening tool that's perhaps a little bit cheaper and automatable, then we encourage you to try VRE 100 and see how it works out for your particular needs.
Thank you very much. Next question will be for Khan. Does trizol RNA isolation have any positive or negative effects on the ability to detect viral RNA?
Actually, this was, I think, tested by Nicolas before.
Yeah, so I did use trizol as a positive control, and it works. Trizol is a very reliable method that's been around for a long time. And so I have used trizol extraction going into RNA, into qPCR. And as long as you, after your precipitation, do the full wash and everything, it should work just fine. But that was, I think, in a relatively clean sample. So I'm not an expert on trizol. I think it has a lot to do with what exactly your sample type is and what other possible contaminants could be in there that would interfere with qPCR. If you have a very clean sample, it should work for qPCR, but not all samples are clean.
Another question for Nick. Is there a maximum amount of time that the sample can be incubated with the viral RNA extraction buffer?
So we tested it up to 30 minutes, maybe 30 or 40, and didn't see any decrease in activity. We didn't see a need to test beyond that because, we figured people prefer faster than slower. But if you wanted to try that, we would say, have the shorter incubation as a control, and you could try storing it in the fridge. But at least up to 30 minutes, you're definitely safe. Beyond that, I would say unknown.
Thank you, Nick. Do the qRT-PCR kits and extraction buffer, do they work with multiplex qRT-PCR as well?
Yes. potentially it's work for multiplex qPCR, but the detailed detailed condition are quite vary based on the combination of the targets, and you need to experimentally test the combination if you want to do the multiplex.
And I believe we have time for one more question, and this one I will open up to both of you, but starting with Nicolas. Can you detect other viruses in addition to SARS-CoV-2 using the extraction buffer, GenElute, or these kits?
Yeah, that's a good question. So with GenElute, we've used similar inactivated particles, and we've seen that with GenElute we can extract and detect RNA from flu B and flu A. With VRE100, we have seen that we can lyse and detect RNA from flu B. We had a little bit of trouble with flu A, so we're still playing around with that. It's unclear if there is specifically a physiological difference with the composition of flu A that makes it particularly difficult. But that's sort of still ongoing. We hope to test RSV soon. So this is primarily for RNA viruses. We've also have a little bit of data with AAV, which is a DNA virus, and it did seem to work, give a little bit of a boost in sensitivity using VRE100. That being said, as was seen from the presentation, really the majority of the data is on SARS-CoV-2 that we have.
Khan, would you have anything to add?
No.
All right. Unfortunately, that is all the time that we have for today. If you have any further questions, please consider reaching out to our speakers directly. Their emails are shown on the screen. As a reminder, the webinar will be archived on the scientist's website, and you will receive an email notifying you when the on-demand version becomes available for viewing. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientist, I'd like to thank our speakers, Nicolas Caffarelli and Saito, as well as our sponsor, Millipore Sigma. Thank you, everyone, and have a pleasant day.